So now we're going to talk about the cell. So cells are what you are made of. Everything that you do, every action your body performs is carried out by a cell. And then, as mentioned, the trillions of cells that make you a human being all work synergistically to maintain homeostasis in your body so that they can also stay alive. Each of these images shows a cell from a different part of the body. Can you identify which came from which? Which one is the muscle cell? Which one is a digestive system cell? And which one is a cell that comes out of your mouth, which is actually a cheek cell? Do you know? So we are going to ask ourselves two essential questions during this presentation. How does the structure of the cell relate to its function? And we're just going to use the standard cell that you were taught in 1406. And what is the relationship between the cell's function or physiology and human illness? So the first question is anatomy and physiology. Cell anatomy relate to its physiology. And the second one is when the cell's physiology is not working correctly, how does that make us sick? To answer which is which, the muscle cells are B, the digestive system cells are C, and the cheek cells from your mouth are A. Okay, so here is the standardized cell that everybody knows. In cytology, cyte is cell, and ology is the study of, so the study of cells. All animal cells have three main parts. Remember, the thing that makes animal cells different from plant cells is that we do not have a cell wall. And the thing that makes animal cells different from bacterial cells is we all have membrane-bound organelles. Y'all remember that, don't you? So we have a nucleus, a membrane-bound nucleus. We have the cytoplasm, which is the jelly-like structure that fills out the cell and supports all the other organelles. The cytoplasm itself is an organelle. And then we have that super important special cell membrane that is a double bilayer of phospholipids to maintain the outside and inside boundaries. All of the body's functions, I already said, are carried out by cells, which are organized into tissues. Remember from orientation of human biology that tissues are all the same cell working together. The organelles are, of course, the Golgi vesicle or the Golgi apparatus or the Golgi body. Those are all things. The cytoplasm itself, a vacuole. Not all cells have vacuoles, but vacuoles are used to maintain pressure in a cell. We have peroxisomes, which are full of peroxide to help break things down. We have secretory vesicles, which means that this vesicle has fused to the cell membrane to release something into the inter or intracellular space. We have the smooth endoplasmic reticulum for lipid synthesis. We have a lysosome for degradation of external things. We have all of the microfilaments and microtubules and intermediate filaments that make up the cytoskeleton. We have the centrosome, which is important in the cell structure, cell cycle, plasma membrane mitochondria, the powerhouse of the cell. Ribosomes for protein synthesis, and ribosomes also embedded in the rough endoplasmic reticulum for protein synthesis. Nucleus, nucleolus, and chromatin. Yes. Okay, so that's everything that you should remember from 1406. Now, as we progress through this PowerPoint, we are going to be referring to this case study. As you see here, this mini case is based on a real person, but all these images are stock images. This is Mackenzie. At 4 years old, she was diagnosed with pneumonia. Her parents were worried. It seems that Mackenzie was always sick. She always had a runny nose and had frequent ear infections. The diagnosis of pneumonia also revealed something else. Mackenzie had a condition known as situs inversus. What do you think situs inversus is? So here we have a child, young child, who is always in some way sick. Runny nose, ear infections, pneumonia, those are all considered upper respiratory infection type things. If you said situs inversus is the condition where all your organs are flipped in your body, you are correct. It is a rare condition, but it does happen. And in fact, Moira, or the woman who plays Moira in Schitt's Creek, who was also the mom in the original Home Alone, she has situs inversus. So there's a famous case there, and others. So instead of your heart being more to your left, your heart is more to your right. Your major organs, liver and stomach and spleen, are flipped. Your intestines are rotated backwards as well. Why do you think this condition can result in problems with the circulatory and digestive systems? So, besides the fact that diagnosis becomes difficult with situs inversus, because if you're experiencing heart pain, you're going to experience it in the wrong side of the body. If you have appendicitis, it's going to be on the wrong side of the body. Those types of things. Typically, situs inversus is not a singular event. It has to do with some genetic abnormalities that could cause digestive system and cardiac defects. Okay, so let's talk. Cells within your body vary in size, structure, and function. Most cells are microscopic. The only human cell of the human body that you can see with the naked eye is an egg from an ovary. That's the only one that's large enough to see without a microscope. But even under a microscope, they all have very different sizes. If you were to look at a blood smear, red blood cells are much smaller than white blood cells, and platelets, which are not full cells but pieces of cells, are even smaller than that. Every cell starts out the same as a stem cell, but the stem cells, when you are being developed in the uterus, are triggered by different hormones and they become differentiated stem cells. And so then you have stem cells that can only create epithelial cells, or you have stem cells that can only create blood cells, or you have stem cells that can only create liver cells. They all have a stem cell. They all come from stem cells, but they are stuck in whatever pathway they are. If you ever get really into the laws guarding human testing and stem cells, the reasoning for researchers to want to always use fetal cells is those fetal cells have not been differentiated yet. So you can turn those stem cells into anything you want them to be that is a cell, right? Because the hormones haven't told them that they are only going to be blood cells, which is hugely beneficial in science and research and medicine, but also comes with all of the emotions and everything about using fetal tissue, which does not have to mean a fetus at all. You can use what are they? A placenta and placental cord cells and still get what you need, but people have ideas. Okay. Here is cells with cilia, and you can see here a goblet cell making mucus. So this is most likely in the respiratory pathway. Here is a giant microbe. If you ever look up giant microbes, there's a whole store that sells these plushies. And this is a neuron where you have the dendrites around the nucleus and then the axon down to the terminal synapses. So you all start out as just a ball of the same cell, and then something in fetal development triggers them to only become neurons, only become blood cells, only become respiratory cells. When we talk about a cell, as you know, that cell membrane is an essential part of the cell, phospholipid bilayer. It consists of phospholipids, integral proteins that are embedded through the lipids, and of course, accessory proteins that are just on one side or the other side of the lipid bilayer. Remember, you can have glycoproteins where glycogens or sugars are bound to the proteins, and you can also have glycolipids, which is not being shown in this picture, where the sugars are bound to part of the lipid membrane, or the cholesterol. Cholesterol is not also being shown in this picture. All phospholipid bilayers include some component of cholesterol. The more cholesterol they have, the stiffer the membrane is, and the fluidity is decreased. And we know that we need these plasma membranes to be very fluid. Because it is a hydrophobic space between two hydrophilic spaces, it becomes selectively permeable, where small non-polar molecules can get through and some water can get through, but not fast enough to keep us alive, which is why we have all these integral proteins to get us through. So what is the relationship, if we're looking at cell membranes, this is referring back to the case study, between glutamine 4, insulin, and glucose? So this is showing you an insulin receptor binding to insulin, and then glutamine brings glucose in to the cell. If something is wrong with any of these parts, you don't get glucose into the cell. So what diseases can this cause? Let's go back. Well, insulin resistant diseases, for one. It's producing insulin that you can't get it into the cell, so that's more of a type 2 issue. Okay, so that's it. So that is a physiology function that is necessary for life. So the doctors, given Mackenzie's history of illness, sniffles, and ear infections, wanted to rule out cystic fibrosis. Cystic fibrosis is a genetic disorder that you are born with, in which your goblet cells overproduce mucus in all of your body systems, and the mucus coats the organs and causes systemic problems. If your lungs are coated in mucus, you're breathing less efficiently. If your digestive system is coated in mucus, you're digesting less efficiently, whatever. So, why is CF considered a disorder related to the membrane? Well, a normal cystic fibrosis transfer channel, I'm going to assume that's what that stands for, moves chloride ions to the outside of the cell. And because the chloride ions are not building up inside the cell but are being evenly distributed, then you have an isotonic solution on both sides of the cell, which is what we want. But if you have a mutant CFTR channel, all the chloride ions stay in the cell, which draws all the water into the cell, which means there's less water outside of the cell, and mucus builds up. Could CF have caused Mackenzie's symptoms? For the runny nose, yes. For the ear infections, actually, yes, for all of it, it could have. But the sweat test, which is a common test to see if there's chloride ions in your sweat, showed that Mackenzie did not have extra chloride. And does this mean that she does not have CF? It's not an exact science, but it is a good indication that she does not have CF. So what's causing this baby's pneumonia? Well, let's look at the cytoplasm, also known as the cytosol, which is the water of the cell. It's not water. Is the area between the nucleus and the membrane that holds and supports all the organelles. Many of the cell's metabolic reactions occur in this area. Think back to cellular respiration in 1406 and how glycolysis occurs in the cytoplasm. It's the necessary first step, and it happens in the cytoplasm. Well, do you remember what metabolism is? That's hilarious. So in the cytosol or cytoplasm is all the organelles, also known as little organ. Organ, and then L's means tiny. That perform specific functions. The nucleus is to the cell what the blank is to a person. If you said brain, you're right, but that's not exact. The cell membrane is to a cell what the blank is to a person. That would be the skin or integumentary system. As we explore each of the structures, consider how a failure in the structure results in disease. How big are cells? There's an interactive, well, let's just click on it. Cell size and scale. So I remember this one. If you look at this, here is, times regular 12 point. Here is coffee, grain of rice. And then you can, scroll and you get smaller and smaller. Grain of salt. There is an amoeba. There's a paramecium. There's that human egg. And then finally we get to the cell sizes. So let's see, skin cell, 30 micrometers. Red blood cell is 8 micrometers. See how there's a difference there? A sperm cell is 60 by 5 micrometers. How much was that egg? Whoops, clicking the wrong thing. Egg is, nope. Oh, 130 micrometers. So see, even in your body, there's quite differences in the size of your cells. Anyway, that's a fun little thing. It's called cell scale. You can just look it up if you want to play. Okay, endoplasmic reticulum. There's two types. There's rough and smooth. They have different functions. The transport system, the whole point of it is to create proteins or lipids that are being sent out of the shell and have to go through the vesicular pathway of the endoplasmic reticulum to a vesicle, to the Golgi apparatus, to a vesicle, to the membrane to be sent out of the cell. Here is a picture of rough endoplasmic reticulum. You can see the hollow parts are the inside or the lumen of the endoplasmic reticulum, and then these little black dots are all the ribosomes embedded on it. Here's the smooth, there is the rough. Smooth ER is for lipid synthesis, and rough ER has ribosomes to make proteins. And so the proteins are created directly into the lumen of that endoplasmic reticulum. Golgi apparatus, we know it to be the post office of the cell. It packages and delivers proteins and exports them in vesicles. It has a cis face, or the side facing the nucleus, and a trans face, which is the side facing away from the nucleus towards the cell membrane. And the vesicles, whatever is being packaged out into vesicles, they are processed through from cis to trans before they're sent to the cell membrane. You can see post office of the cell, adorkable. Oh my goodness. And then it's sent on its happy way. Mitochondria, or if you want to be fun, mitochondria, but don't do that. It's the powerhouse of the cell. It's the only thing you probably remember from high school biology. This is where, through cellular respiration, we create ATP by breaking down glucose completely, and we use oxygen to do that. Remember, the reason we use oxygen to do that is the very last step of the electron transport chain is to put high energy electrons back onto an oxygen and create water. That's why oxygen is essential to our lives. The mitochondria produces ATP. What I learned in cell bio is the mitochondria is power cell. Do any of y'all know how to write a check? No, but you know this, right? Okay. Okay, so mitochondria is something that, if they aren't functioning correctly, can cause systemic diseases. Mitochondria create energy. There are a lot of disorders, human disorders, that have to deal with mitochondria not functioning correctly, and they would all end up with like low energy as being one of the primary signs. Cells lack energy to perform basic life processes. So skeletal muscles will be weak, heart won't pump effectively, the eye won't have the ability to move, the liver won't be as effective, or the kidney as effective as filtering, pancreas won't produce insulin, blood won't be created fast enough, or they'll be created and be wrong. Your inner ear can't hear, your colon will not push food through and defecate properly, and your brain, seizures, myoclonus, which is myoclonus is when your muscles kind of seize up on their own. Well, your brain is not sending the right signals to them. Ataxia is the inability to walk, stroke, dementia, and migraine. You can see here is an individual suffering from mitochondrial illness, unfortunately. Anyway, Mackenzie was tested, and doctors found that her mitochondria were functioning properly. So that's great. we're gonna watch this video. It's a 5-minute video. The reality of mitochondrial disease. Oops. Well, I hope you all did enjoy that musical interlude there. If you notice, a majority of the children that have been diagnosed with these mitochondrial diseases or disorders, sorry, not a disease, have feeding tubes, and that is because their digestive systems are not working properly and they need help in digesting. Okay, so Mackenzie was tested and we found that her mitochondria were functioning properly. So then we have lysozymes. Lysozymes are sacs full of hydrogen ions, which are like free radicals, which are we all know are really bad for us, but they're sequestered away into a vesicle, and they are used to break down digested material that needs to be utilized by the cell or to break down old things in the cell that they don't need anymore. It's called, well, I don't like that term, but that's fine. It breaks down substances in the cell, such as food or worn out cell parts and even bacteria. And you can see in this nice little um, Amoeba Sisters GIF how it works. What organ in your body is most like the lysosome? Ah, yes, the stomach. Okay, so lysosomes have their own storage disorders, and there are hundreds of them based on what exactly is wrong. One of the most common ones is Tay Sachs, which is a progressive deterioration of nerve cells that leads to mental and physical disabilities that begin around 7 months of age and usually results in death by the age of 4. The reason being, if you cannot break down old proteins or things that are no longer needed by the body, they just build up in the body, eventually slipping into the bloodstream because there's nowhere else for them to go. Protein buildup in the bloodstream is detrimental to all of our soft organs, and it causes brain damage and it causes kidney damage and liver damage and all those things, which results in a much shorter lifespan. Do you think Mackenzie has a problem with her lysosomes? And this information right here, you kind of already know the answer to that because it usually results in death by the age of 4. And how old is Mackenzie? That is right, she's 4. Okay, so centrosomes. So what? Whoa, let me try that again. Centrosomes, or centrioles, whatever, are two cylinders that are perpendicular. They are specialized motor proteins that we have that are two microtubules surrounded by 9 sets of microtubules. And they form that super special mitotic spindle to create metaphase and to separate the chromosomes during anaphase. And the spindle moves chromosomes during cell division so that each new cell gets the proper number. And then also made up of motor proteins is our cytoskeleton, made of microfilaments, intermediate filaments, and microtubules for cell shape and support. This can also include cilia, which are short structures that beat the fluid around them and create movement, or it can include flagella, which are incredibly long comparatively to the cell that are for purposeful movement. Doctors wanted to test Mackenzie for a disorder called primary ciliary dyskinesia. This means difficult, painful, or bad. So in this case, it would be difficult. Kinesi will be movement. So like kinesiology, the study of the body movement. And ia is pertaining to. So pertaining to difficult movement of the cilia. This disorder causes cells to not create normal cilia. Where do we have cilia in our human body? We mostly have it in our respiratory system. You have it in your nose, part of your trachea. You have it in your bronchioles. And the whole point is to move trapped things in the mucus up or down to the mouth so that it's swallowed and get digested in the digestive system. So here is a cell scraping that shows what her cilia look like versus what normal cilia look like. And you 1, 2, 3, 4, 5, 6, 7, 8, 9. Wait, 1, 2, 3, 4, 5, 6, 7, 8. Yeah, 9. We have 1, 2, 3, 4, 5, 6, 7, 8, 9. But they're mutated somewhat. They're not as clear. And then, of course, you're missing the very essential 2 in the middle. Those are the central microtubules. They are gone. What would problems with cilia cause? Pneumonia, ear infections, and a constant runny nose? Because you can never ever get the bacteria out of your respiratory system. So, Mackenzie has primary ciliary dyskinesia, which is common with situs inversus, but she will always have a cough and deal with lung and ear infections. So, in utero, when your cilia are not created correctly, we believe that cilia may play a part in determining the placement of organs within the thoracic and abdominal cavities. If your cilia are not working correctly, you have a 50-50 chance of having situs inversus. Wild, huh? Doctors have warned her parents that when she gets older, she may have trouble conceiving a child because you also have cilia in your fallopian tubes and at the end of your fallopian tubes called the fimbriae that move the egg into the uterus, and is at risk for ectopic pregnancy because if that egg is fertilized outside of the uterus, which can happen, the egg will implant anywhere and it can be somewhere in the abdominal cavity. And I just explained what role it plays. So then you have the nucleus, the brain of the cell, directs genetic activities, and it keeps the DNA in the form of chromatin. The whole point of the nucleus and the DNA is to make proteins to function in the body. Nucleolus makes some ribosomes, and the nuclear envelope has pores so that proteins can leave through, mRNA can leave through the pores and create proteins. So you can, more analogies, what part of the cell is most like the stomach, circulatory system, brain, bone, skin? Y'all answer that on your own. The animal cell, and here's your animal cell again with all the structures that you know. Now, you need to test yourself that you can still label all of these correctly. The big picture is that the cell makes a protein. It's a factory by the instructions of the DNA, and each part of the cell has a job to do. As stated, cells are microscopic. This is cheek cells. You can only see the nucleus and basically the plasma membrane in a light microscope at 40x, but it's 400x because the oculars also have a 10x magnification. We're not going to do the cheek cell lab because we're not in person and you have to have an iodine stain that y'all don't have access to. Movement of substances across the membrane. This is 1406. I'm not going to take too long. You know that it is a semi-permeable membrane, and cells are limited in size because we just don't have that high of a diffusion rate to be any bigger of cell size. Diffusion is molecules moving down their concentration gradient from high to low. If it does not require energy, this is passive transport. If it's too big to go through the cell membrane on their own, it has to go through a integral protein. This is called facilitated diffusion. And then there's active transport where you have to use ATP to get something across the cell membrane. Osmosis is that diffusion of water through a selectively permeable membrane. The reason they don't use pure water in an IV bag, that's right, we are not made of pure water. We are saltwater organisms. If you create pure water, you create a hypotonic solution, and all that water flows into the cells and explodes them. Do y'all remember hypo, and isotonic solutions? I hope so. And here's a semi-permeable membrane. If it's isotonic, everybody's happy and water is flowing in at the same rate it's flowing out. If it's hypertonic, there's too much salts around the solution of the cell, so water moves out of the cell to try to diffuse those salts away, and the cell crenates. That can be saved. If it's a hypotonic solution, which we would create with pure water, all the water moves into the cell because it has more salts inside. Very, very little water comes out. The cell swells, and then because we don't have cell walls, it bursts, and you cannot recover from that. And here is once again those lovely gifts from Amoeba Sisters. Okay, active transport requires energy, we said. And then there's the life cycle of the cell where we take the chromatin, we duplicate it, no, we don't. I don't know. Oh, my land. We duplicate it, we separate it, and we make two new cells. It's not my job to teach you prophase, metaphase, anaphase, telophase. You should already know how all that works, the whole PMAT process. So if you don't, you need to go remind yourself of how it works. Cytokinesis. So that video did a very good job. Here is prophase, sort of metaphase, but they're not lined up very well. Anaphase and telophase. Yes. Okay, cells. Stages of mitosis. Right. What is the relationship between cancer and mitosis? So this is when mitosis goes wrong. It is the unstoppable creation of new cells, and a whole bunch of cells that you don't need becomes a tumor. It can be benign, which means it's harmless, or it can be malignant, which means it's harmful, but it is what we call cancer. Oh my God, stop it. Stop dividing. You're making a tumor and you'll eventually kill us all. Just following orders. And the orders are the DNA telling it to just make more because checkpoints have failed. Apoptosis is something that happens in your body every single day, because I don't want to alarm you, but we are creating cancer cells every single day, and the apoptosis comes along to stop that from happening. We are thankful for apoptosis, and it's called programmed cell death, and it's when your own immune system tells your cell it has to die because it's causing a problem, and the cell just goes, sir, yes, sir, and does that. This is malignant melanoma. It's, yes, the most aggressive form of skin cancer, according to my brain. The reason cancer becomes so bad is that once a tumor becomes large enough, they start breaking into the lymphatic system and the blood system, and then you start building tumors everywhere in your body, and that pushes against your normal organs, and it causes disruptions in the organ's ability to function. And if that happens enough, it leads to death. It's not great. Normal cancer. Cervical cancer and HPV. I don't know how old y'all are, but I hope you all have had the HPV vaccines, male and females. It's one of the most common causes of death in females, and it's one of the easiest things we can prevent. And if you don't know about HeLa cells, this is Henrietta Lacks. HeLa is the abbreviation of her name. Henrietta Lacks died from cervical cancer in 1951. Oh, yeah. But the cells harvested from her cancer tumor are still alive to this day, and they are the main cell line for research that we have. But these cells were harvested and used in research without Henrietta Lacks or her family's permission. They do not die after a set number of mitosis like most cells do, and so they can keep these cells indefinitely. So even though Henrietta Lacks is dead, she's actually still literally alive on this planet. It's kind of terrifying. HeLa cells were used to develop polio vaccines. They have been sent into space. They are used in HIV/AIDS research. Basically, anything with pharmaceuticals is done on HeLa cells. It was not ethical. We know now historically that African Americans, Native Americans, anybody, of course, but white people, have been systemically treated poorly by the medical community. Her cells were obtained without anybody's knowledge, and they were used without anybody's knowledge in pharmaceuticals. Should scientists ask for consent before using someone's cells? These are ethical questions. There's really no right or wrong answer in ethics, but you can decide for yourself. And does the patient or family deserve compensation for the use of the cells? Depending on whether you believe yes or no, her family actually has been compensated. It happened about 2 years ago that they were finally like, hey, sorry, we did this. Here's a little bit of money. And of course, Oprah, that's her name, she was in a movie about Henrietta Lacks. And then here's a self quiz. So I am not going to give you the answers, but here's question number 1. Question number 2-ish. Question number 3. Ooh. Any of y'all can read. Don't want to guess at what that writing's actually called. I'll offend somebody. Structure. Name. What are we looking at? That's a very poor slide. Structure and functions. Well, it gives you that. Okay. Which beakers contain a solution that is hypertonic relative to the bag? What will happen to the baggie in the hypertonic solutions? Can you name these? The end. I appreciate your time. Thank you so much.